Um, Biology, Chapter 12, Video 8, Theory. Now we have learned that we discussed that most of the energy is made available as a result of respiration by a process that involves transfer of electrons. So now we are going to deal with that part. Oxidative phosphorylation and the electron transport chain. In the final stage of aerobic respiration, so now up until this point, the involvement of oxygen was quite limited. In Krebs cycle, there was no involvement of oxygen at all. So now here is the point where oxygen comes along. In the final stage of aerobic respiration, oxidative phosphorylation, the energy for the phosphorylation of ADP to ATP, that is, phosphorylation is the addition of phosphate, comes from the activity of the electron transport chain. Oxidative phosphorylation takes place in the inner mitochondrial membrane. So it happens in the inner mitochondrial membrane, right? So now let's look at this diagram. You might not understand the events that take place here, but after we have discussed everything, you would be able to get a clear idea as to what happens here. So you have this reduction of NAD. You have a reduced NAD. That reduced NAD will actually become NAD here. And in that process, H plus is given out, which is released to the outside, and an electron is transferred. That the same thing happens with the reduced FAD as well. So this thing is, as you can see here, it happens in a part called the respiratory complex. So this is a respiratory complex. We will understand why they are known as such and what exactly happens there. So these electrons get transferred here, and then it combines with high H plus that comes through the membrane and joins with oxygen to produce water, right? So this particular thing, this particular process happens inside the inner mitochondrial membrane. And let's see how the events unfold here. Right. Reduced NAD and reduced FAD. So I hope you can remember from where they come. They are produced as a result of the Krebs cycle. Reduced NAD and reduced FAD are passed to the electron transport chain. So that begins there. Here the hydrogens are removed from the two hydrogen carriers, and each is split into its constituent proton and electron. So the hydrogens are removed from the hydrogen carriers. So these hydrogen carriers are NAD and FAD. So from them, hydrogen is removed. This hydrogen will actually it will go out. It will be split into the H plus and the electron, or the proton and the electron. The energetic electron is transferred to the first of a series of electron carriers. Most of the carriers are associated with membrane proteins, right? So we are talking about the energetic electron is transferred to the first of a series of electron carriers. Don't confuse it with the hydrogen carriers, okay? So the most of the carriers are associated with membrane proteins, of which there are 4 types. A functional unit called a respiratory complex consists of one of each of these proteins, arranged in such a way that electrons can be passed from one to another down an energy gradient. So now, in here, as you can see, this respiratory complex, there are 4 types of proteins, and they will get transferred in a certain manner, right? You don't have to actually know the entire detailed structure of how the electrons get transferred, but you need to know that it happens in such a way that it goes through an energy gradient. It goes down an energy gradient. As an electron moves from one carrier at a higher energy level to another at one at a lower level, energy is released. Right? So when this electron moves from one carrier protein to at a higher energy level to another carrier at a low energy level, energy is released. Some of this energy is used to move protons from the matrix of the mitochondria into the space between the inner and outer membranes of the mitochondrial envelope. Right? So this energy, some of this energy is used to move protons from the matrix of the mitochondrial matrix into the space between the inner and outer membrane of the mitochondrial envelope. This produces a higher concentration of protons in the intermembrane space than in the matrix, setting up a concentration gradient. So this part you should understand as a story. Step by step you have to understand. There is nothing much to understand. You have to remember. You have to remember these parts as a story. Now protons pass back into the mitochondrial matrix through protein channels in the inner membrane, moving down their concentration gradients. Now protons left the inner matrix and entered the intermembrane space. So there are more protons there. Now what happens is, because the concentration is higher, more protons will come back into the matrix through protein channels in the inner membrane, and they move down their concentration gradient. So as you can see here using energy, they are being these hydrogen ions are being moved out. Right? You can see that. But because there is a concentration, because the concentration of hydrogens here increase, so the protons increase, H plus are protons. There is a higher concentration of protons there, and as a result, what happens is hydrogens begin to move in. They begin to move in from here through the channel proteins. They come in. All right. I hope you understood up until that point. Now protons pass back into the mitochondrial matrix through protein channels in the inner membrane, moving down their concentration gradient. Associated with each channels is the enzyme ATP synthase. So as you can see here, in this channel, at the end of the channel, you have ATP synthase. Right? So that is the enzyme involved in or responsible for the production of or synthesis of ATP. As the proton passes through the channel, the electrical potential energy is used to synthesize ATP in the process called chemiosmosis. Right? We discussed what chemiosmosis is. Finally, oxygen has a role to play as the final electron acceptor in the mitochondrial matrix. So as the H plus comes in, or the proton comes in, their electrical potential energy, because there is a potential difference between outside and inside, that electrical potential energy is used to synthesize ATP, and that process is called chemiosmosis. Then oxygen comes in as the final electron acceptor. In the mitochondrial matrix, an electron and a proton are transferred to oxygen, right? And an electron and a proton are transferred to oxygen. As you can see in this part, that is exactly what happens in this part. An electron and a proton is transferred to oxygen. and that results in the production of water. Now the process of aerobic respiration is complete. The sequence of events in respiration and their sites are shown in figure 12.11. The balance sheet of ATP. So let's go to 12.11 and see how they are, where they are occurring. So as you can see, we start off with glucose here. We start off with glucose and it goes through glycolysis, and that produces pyruvate, and the pyruvate goes through link reaction to produce acetyl coenzyme A. Acetyl coenzyme A goes through Krebs cycle to produce 1 ATP, and then reduce NAD and reduce FAD comes in. So as you can see, this the glucose and glycolysis process takes place outside of the cell. Outside of the cell, in the matrix. Apologies, please disregard the lines that have been drawn. So the process, this part happens outside of the cell, that is in the matrix. This part, this is the in nuclear, but this is the mitochondrial envelope, right? So I hope the diagram is clear. And then this reduced NAD and this part, the link reaction and Krebs cycle take place inside the mitochondrial matrix, and then reduce NAD and reduce FAD, they give away the H plus and the electron. Then that electron transport chain happens inside the proteins, that the carrier proteins, the electron carriers, and that takes place, electron transport train and the oxidative phosphorylation takes place inside the mitochondrial matrix. Right, so that is those are the details where it is taking place, and you have to have a clear idea on this. Try to memorize this chart. And now the sequence of events in respiration and their sites are shown in figure 12.11. The balance sheet of ATP used and synthesized for each molecule of glucose entering the respiration pathway is shown in table 12.1. So this is table 12.1. In glycolysis, 2 ATPs are used and 4 are made, so net gain is plus 2. Link reaction, there are no ATPs being produced. Krebs cycle, 2 ATPs are made. Then oxidative phosphorylation, 28 ATPs are made. So all-together, 34 ATPs are made and 2 are being used in glycolysis. So the net gain in ATP is 32. Theoretically, 3 molecules of ATP can be produced from each molecule of reduced NAD.